So today I will be talking about metabolic therapies. And metabolic therapies can include dietary therapies, nutritional supplements, and also various drugs that can influence energy metabolism in a way that causes a therapeutic effect. So my talk today will focus more on dietary therapy, which I think you're familiar with, ketosis or the ketogenic diet, and also ketone supplements that can mimic the effect of the ketogenic diet without the dietary restriction. So there's many therapeutic implications for metabolic therapies. They can influence a broad range of pathologies because in one way or another, most diseases, especially diseases of the brain, neurodegenerative diseases, are linked pathophysiologically to energy dysregulation. So a metabolic therapy can often enhance brain energy metabolism in the face of these metabolic deficits. And the last half of my talk will be more on the practical application. So what can you take away from what I'll show a lot of science up front, and towards the end I want to talk specifically about applying these things through your diet and through supplementation. And I have a list of, I can only talk to a certain extent based on time, but I have a list of resources that I think will be helpful for you. So my research on metabolic therapy evolved from funding from the Office of Navy Research. So the Office of Navy Research has been funding me for 8 years now to develop and test strategies to prevent central nervous system oxygen toxicity seizures. So the first part of my funding as a postdoctoral fellow was to specifically study the cellular and molecular mechanism of oxygen toxicity seizures, which occur in Navy SEAL divers that breathe 100% oxygen or high concentrations of oxygen with a closed circuit rebreather. The development of a mitigation strategy to prevent oxygen toxicity seizures had therapeutic implications for a wide range of things, including neurodegenerative diseases. Our lab studies ALS, Alzheimer's disease, Angelman syndrome, and a variety of different seizure disorders. Also has implications for wound healing, and as Ken mentioned, cancer. So oxygen toxicity, or central nervous system oxygen toxicity, are seizures that result from breathing oxygen at hyperbaric pressures. So typically pressures above 2.5 atmospheres absolute, which is roughly about 50 feet of seawater. So CNS oxygen toxicity seizures are a limitation for hyperbaric oxygen therapy, which is used for about 14 different clinical applications. It's FDA approved for. This includes some like decompression sickness, carbon monoxide poisoning, ischemic wounds. A variety of things can be treated with hyperbaric oxygen therapy. A limitation of exploiting the benefits of hyperbaric oxygen therapy is the risk for these oxygen toxicity seizures. So, and importantly, as it relates to my ONR funded project, diving, specifically with a closed circuit rebreather, special ops divers, Navy SEALs, will use a closed circuit rebreather which has roughly 100% oxygen or a high concentration of oxygen, and it limits their ability to dive deep. For example, with the exposure limits, diving down to 50 feet of seawater, you'd have the potential to get oxygen toxicity seizures in just 10 minutes. And this can vary tremendously between different individuals. It can vary even in the same individual. If the individual is under psychological or physical stress, the potential for oxygen toxicity can dramatically increase. But it's highly variable between individuals. So we wanted to understand fundamentally, what's causing these oxygen toxicity seizures. We needed to know the cellular and molecular mechanism of CNS oxygen toxicity. So it's not known. There's no way to predict these seizures when they occur, and there's no way to prevent them as of now. But to really understand fundamentally why it's happening, we needed to know the cellular and molecular mechanism. To do this in our lab, we use a variety of methods, including atomic force microscopy, which gives you a very high-resolution image of cells that you're looking at. And it also has the ability to physically interact with the sample. And it gives you the resolution of an electron microscope, but you can actually visualize living tissue with it. So it's a very effective tool for understanding how this works. Laser scanning confocal microscopy, fluorescence microscopy, these techniques allow us to look inside of a cell and optically section the cell to look at the organelles. For example, the metabolic therapies that we're working on really target mitochondrial function. So to enhance and preserve mitochondrial function under conditions of environmental extremes, if we can preserve energy metabolism in the brain under these environmental extremes, we think we can protect the warfighter in the environment. Electrophysiology, we can record directly from neurons in the brain, and radiotelemetry. And what we've done is adapted these techniques over the years to hyperbaric chambers. So we're able to take off-the-shelf equipment and install them inside hyperbaric chambers and simulate a dive inside the chambers. So this allows us to, using atomic force microscopy, it's essentially a scanning probe that raster scans back and forth, and it gives you information on the cell membrane itself, and it allows you to visualize or capture images of the proteins even. And you can functionalize the tip to push down and up on the membrane to look at changes in the viscoelasticity of a membrane. So we think the membrane fluidity changes in response to the level of oxygenation over time. And as these lipids, which are polyunsaturated fatty acids, as they become oxidized, it changes the fluidity of the membrane itself, and also the proteins that are embedded in the membrane. And these proteins play a big role in the cellular function or electrical excitability of the brain. So we think they're imparting a toxic effect at high levels of oxygen, or the free radicals that are generated with high levels of oxygen are changing the proteins in the membrane. So with scanning confocal microscopy, we can look inside the cell. In this image here, we can measure oxygen-free radicals in real time under levels of hyperbaric oxygen, and we can image the mitochondria inside the cells. So we can look at mitochondrial membrane potential, mitochondrial superoxide production, which is a precursor to free radicals. And at hyperbaric pressure, we can also measure, using EEG and a variety of physiological tools, we can measure seizure generation under hyperbaric conditions. So using these tools, what we've been able to do is to develop a model of the mechanisms of CNS oxygen toxicity. So there's a lot of science up front here, so I'm just going to kind of gloss over it, but I think it'll give you an appreciation for kind of later on how our metabolic therapies are working. So we know that if we increase the level of oxygen in the brain, there's a proportional increase in the level of something called superoxide anion. And this is a free radical. We're all familiar with free radicals. They're also called oxygen free radicals or reactive oxygen species. And in our experiment here, we actually measured in real time the production of oxygen free radicals in the hippocampus. The hippocampus is an area of the brain that's important for learning and memory, and we think it's an area of the brain that's involved in seizure generation, whereas seizures it's a very oxygen sensitive region of the brain. If you have a stroke or you're deprived of oxygen, it's an area of the brain that's selectively vulnerable to hypoxic injury. So when we hyperoxygenate the brain, the level of free radicals skyrockets in this area of the brain that's very critical for learning and memory. And we actually measured the production of oxygen-free radicals over time, over 4 hours, in response to graded levels of oxygen from 20 to 95% oxygen. And we've shown that with increasing levels of oxygen, there's an increase in cell death too. So with the production of superoxide anion, there is, I'll just say there's an increase in more reactive oxygen species, bless you, that like hydroxyl radical. And these can go on to cause membrane lipid peroxidation. And ultimately, it's influencing the membrane itself. Here's an image of using atomic force microscopy where we can measure, typically the membrane is smooth, but we start to see it blebbing up under conditions of high levels of oxygen. So we think this membrane damage is influencing the electrical activity that we call it the membrane potential across the cell membrane. And it's that membrane potential that really drives a lot of the cellular processes essential for life. So a dysregulation of the electrical activity across the membrane can ultimately lead to hyperexcitability. And using electrophysiology under hyperbaric oxygen conditions, we can measure in the hippocampus, this is an example, we can measure electrical activity. And we get an increase with hyperbaric oxygen conditions, an increase in the amplitude of what's called the orthodromic population spike. We stimulate here, and then we record on the other side of the region. And under conditions of hyperbaric oxygen, when you stimulate on one side of the brain, you get a greater response on the other side. So the brain is hyperexcitable. And we also see these secondary spikes that are analogous to a seizure in an in vitro preparation. So we can actually measure seizure or seizure-like activity in a petri dish, essentially, in our experimental preparation. In addition, we can do animal studies where we can push the limits of oxygen to generate a seizure. And these seizures are actually quickly reversible and not harmful to the animal once we flush in just air and flush out the oxygen. So we have a variety of tools that allow us to develop a model to better understand CNS oxygen toxicity. So the strategies to prevent CNS oxygen toxicity are very limited. And the Navy has very conservative guidelines to dive within the exposure limits. So at 50 feet of seawater, they're limited to 10 minutes. And if you're in situations where, and I've read about many of them, if a Navy SEAL is diving and they're taking, fire from above with a 50 caliber machine gun and they have to dive down or plant a mine, so there's many different scenarios where they need to exceed these exposure limits to fulfill the operation. So it's a major limitation to their diving operations. So staying within the exposure limits is really the only strategy they have to prevent it. There's been experiments by several groups using antioxidants, and these antioxidants, they work really well in vitro in scenarios where you have cultured cells or brain tissue. But when you take antioxidants and you do experiments with animals, they don't work in an in vivo situation. Anti-epileptic drugs are one potential way to prevent these oxygen seizures, but they've proven largely ineffective in animal models. And it's not to the benefit of the warfighter to take an anti-epileptic drug because there's a host of bad side effects that can hamper performance, cognitive and physical performance. So interestingly, in the literature, I came across a body of literature where multiple groups have demonstrated that starvation can delay the onset of CNS oxygen toxicity by over 200%, up to about 250%. So this was remarkable considering even the best anti-seizure drugs were incapable of delaying oxygen toxicity seizure to that extent. So the question we asked, or that was really on my mind, is how does starvation change brain energy metabolism? So we've known for years, for many years, that fasting is a way to cure epilepsy. There's been reports dating back, this dates back to 1922. And I mean, it's going back to the time of Hippocrates in 400 BC. So fasting was a cure for seizures. It's been known for a long time. So much of what we know about fuel metabolism or brain metabolism during starvation actually comes from the work of George Cahill at Harvard Medical School. And George Cahill, the late George Cahill, he recently passed away about a year or two ago, but his group performed really groundbreaking work to demonstrate, the first paper came out in 1967, to demonstrate that the brain can use ketone bodies as a source of energy. Prior to the study at Harvard that was done in 1967, it was thought that the brain could only use glucose exclusively as an energy source. So he did a very interesting study where he had medical students that were overweight. They fasted for 40 days, and it was observed that their blood ketone levels after about a week skyrocketed and actually exceeded the levels of glucose. And essentially what he showed is that ketones become the predominant fuel for the brain and peripheral tissues during starvation. So in our fed state, if we're eating carbohydrates or a normal diet, about nearly 100% of our energy, brain energy, comes from glucose. But in a state of starvation, it's been shown that about 2/3 of our energy, or even more, are derived from ketone bodies. So ketones function as an alternative fuel for the brain when glucose is not available. So how can we exploit this, right? So starvation's not a very feasible approach to oxygen toxicity, right? So what's interesting is that, and you've heard about the ketogenic diet before, that the ketogenic diet can mimic fasting in that physiologically it can elevate blood ketones and lower blood glucose, and you get a profile that's similar to a person who has fasted. So we know that getting into a state of nutritional ketosis involves an uncomfortable degree of carbohydrate restriction for many. So you really have to restrict carbohydrates, and you have to eat a significant amount of fat to elevate blood ketone levels. So free fatty acids, or fat from our diet, and the mobilization of free fatty acids from our adipose tissue, from our body fat, are processed in the liver through beta oxidation and form these ketone bodies. But we need to deplete the liver stores of glycogen to a certain degree, and we need to suppress insulin for our bodies to be able to make, for the liver to be able to make these ketone bodies, which are a very efficient, high energy alternative fuel, especially for the brain and the heart, and also for peripheral tissues like the skeletal muscle and the skin. So this is very difficult to sustain, and we know that the ketogenic diet is used for drug refractory epilepsy. When kids are resistant to anti-epileptic drugs, or they become refractory to them, the ketogenic diet works when drugs fail. So it's actually become like the standard of care when drugs fail to treat difficult seizures. And it's really grossly underutilized approach because it's so effective, but it's still, just because doctors are not educated in the diet, and also with compliance, it's hard to follow. It's not prescribed too often. So it's difficult to sustain, and for the use in oxygen toxicity, it takes like 24 hours to really even get into ketosis. So it's not very practical for a warfighter to get into ketosis prior to a dive. So I became interested in the idea of ketone supplementation. And there's a variety of ketone supplements that are on the market, or shall we say ketogenic precursors. And the one you may be familiar with is medium chain triglyceride oil, or MCT oil, which is derived primarily from coconut oil or palm kernel oil. And MCT oil, when taken orally, can elevate ketones up into a mild, a fairly low range, but it's still having a significant effect. There's even a double-blind placebo-controlled study showing that it can improve cognitive function in people with mild cognitive impairment. So it's a fat that has a functional effect on brain metabolism. More recently, there's been the development of ketone salts. These are pure ketones in the form of a mineral preparation that can be taken, and it can be used to elevate blood ketone levels. Our research has focused primarily, we do research with these naturally derived agents, but most of my research is involving synthetic agents called ketone esters. And there are ketone esters of beta-hydroxybutyrate and acetoacetate are the two ketone bodies that your liver makes, that your brain uses and your body uses as alternative fuels. So we've been able to develop these synthetically, and they break down to the natural ketone bodies that normally occur during the ketogenic diet. A single administration of a ketone ester causes a rapid and sustained elevation of blood ketone levels within 30 minutes and occurs and sustained for over 4 hours. And we've done studies now up to 12 hours. We can sustain ketosis with a single administration of both. The ketone ester that we chose for our study elevates both beta-hydroxybutyrate and acetoacetate. And beta-hydroxybutyrate is the primary ketone body that you measure in a blood test, but we find that acetoacetate is very important for seizure control. So seizure control correlates more with the elevation of acetoacetate. So therapeutic ketosis up to 2.5 millimolar is difficult to sustain, but with dietary ketone supplementation, we found that it's possible. So we can deliver ketones within 15 to 30 minutes with administration of these synthetic ketogenic agents. So in this case, this is ketone supplementation, an acute dose within 30 to 60 minutes interestingly causes a decrease in blood glucose levels. So we find that elevating ketones can reduce levels of blood glucose similar or even above what you could achieve with something like metformin. So our lab also uses anti-hyperglycemic agents like metformin, and we've done some work with that. So we can suppress glucose and also elevate the 3 ketone bodies, including beta-hydroxybutyrate and acetoacetate, which breaks down to acetone. And we use a variety of techniques to measure ketones, including blood and urine. Acetoacetate is a way, and there's also a breath analyzer that can detect acetone. So more a non-invasive method. So more importantly, we found that these ketogenic agents, or therapeutic ketosis with a ketone ester, delays central nervous system oxygen toxicity in rats. So elevating ketones within this range, our experiment was set up to give a single administration of a ketogenic agent and then dive the rat into 132 feet of seawater, which is 5 atmospheres absolute. This typically causes a seizure within about 10 minutes. And in our rat models, but with the ketone ester administration, we can go over an hour. In almost every scenario, the rodents were able to withstand. It gave resilience against this environmental extreme to well over an hour. And we've demonstrated that we have a 575% increase in seizure resistance, which is above and beyond any kind of anti-seizure medication that has been tried in this model. So we're really encouraged that we've developed a metabolic therapy that is essentially a form of energy, a food, if you will, that you can take orally that gives the brain a source of energy that allows the brain to withstand, and it preserves brain energy metabolism in the face of the oxidative challenge from the hyperbaric oxygen. Now we think that this would also work for a variety of conditions associated with impaired glucose metabolism. So the next question we have is, what is the anticonvulsant and neuroprotective mechanism of ketones? I know this looks scary, but I just want to focus on this here for right now. And what we're doing is, it's called metabolomics. And we can take sections of the brain or the blood, and we can look at 3,000 different energy metabolites in the blood. And this gives us a snapshot as to what's going on, how giving a metabolic therapy alters the global metabolic state of the organism. And what we found, interestingly, is that in some of the studies we're doing now, that glutamate is an excitatory neurotransmitter and is used to produce something called GABA aminobutyric acid, or GABA, which is inhibitory. And the ratio of glutamate to GABA will sort of dictate the excitability of your brain. So you have an excitatory neurotransmitter and inhibitory, and the ratio of this changes with administration of a ketogenic diet, but also we found more recently that it changes with ketone supplementation. So there's the GABA to glutamate ratio is higher when you're in ketosis. So not only does it deliver more energy to your brain, it puts the brain into, it prevents the brain from being in a hyperexcitable state that could trigger a seizure. So our research is also looking into the production, which I showed you images of reactive oxygen species, also known as free radicals. And we find that ketones can suppress the generation of free radicals through a variety of mechanisms, we think by enhancing mitochondrial function. So the mitochondria are the energy organelles within the cell, and they're also the sites of free radical production. By enhancing mitochondrial efficiency, you prevent, as oxygen goes through the mitochondria to create energy, less of that oxygen becomes an oxygen-free radical, which could go on to damage. So ketones tend to be, their neuroprotective mechanism may work in part through a reduction of reactive oxygen species. But I think, more importantly from my perspective, I think there's an increase in energy reserves of the brain. So if we look at these, it's called tricarboxylic acid metabolites, we see that they all go up, and basically showing that the brain reserves of energy are going up. So there's enhanced brain energy metabolism. So how can we exploit the therapeutic effect of ketones? We think that ketones, being in ketosis, will create resilience not only against hyperoxia or hyperbaric oxygen, but also during potentially hypoxia, which could occur during a stroke or during a ischemic event, or even under hypobaric hypoxia or altitude. As we climb in altitude, spaceflight, the pressure goes down, and this creates hypoxia in the brain. And we think there'll be ketones enhance energy production under low levels of oxygen, so you derive more energy per oxygen molecule. We think it'll be helpful there. Traumatic brain injury, Parkinson's disease, these are all areas where I think ketones can be therapeutic. So our lab is studying right now, and I won't have time to go into these, but we're looking at the application of therapeutic ketosis for Lou Gehrig's disease, also known as ALS, Alzheimer's disease. There's been a clinical trial that started at my university to look at this. They're using a naturally derived ketogenic agents. Angelman syndrome, which causes seizures and motor dysfunction in kids, and different epilepsy models we're looking at right now. And we find that with ALS, Alzheimer's, Angelman syndrome, in our animal models of disease, the motor function is off the charts. So the animals run faster on a treadmill-like apparatus, and we see a lot of encouraging results, preliminary results in our studies. Glucose transporter deficiency syndrome is a syndrome where a genetic defect causes a downregulation of the glucose transporter in the brain. And the kids who have glucose transporter deficiency, their brains are literally starved of glucose. So they need to stay on a ketogenic diet to actually prevent seizures and to prevent them from being totally immobile. So the ketogenic diet needs to be very strict for them to be able to function. So delivering a ketogenic agent like a ketone ester could nourish the brain without the dietary restriction. So we see a really big potential application there. With wound healing, in many cases, impaired wound healing occurs in older patients or smokers where you have high blood glucose and an impairment of blood flow to the wound area. Ketones have been shown to increase perfusion of blood into the area. So we have some really encouraging data in our wound healing model using oral forms of ketones. So I'm going to focus the rest of the talk on cancer. And my work started on cancer after some observations that we made in cancer cells. So when we developed these various technologies that I showed you earlier, like atomic force microscopy and scanning laser confocal microscopy, we looked at a variety of cells to just curiously, we wanted to know what do cells do under hyperbaric oxygen, like what happens to cells. So we looked at muscle cells, we looked at fibroblasts, skin cells, and we also looked at various cancer cell lines. And we observed that high oxygen is toxic to cancer cells. So it causes an overproduction of oxygen-free radicals, which overwhelms the antioxidant capacity of the cells, and it causes them to die. And oxygen toxicity happens in healthy cells, but cancer cells were selectively vulnerable to the level of oxygen that was relatively non-toxic to healthy cells. So they were just more vulnerable to the oxygen toxicity. So, and we published this finding, but I didn't really follow up on it until a year or two later. We also demonstrated that ketones were toxic to cancer cells. So your normal cells, even in a culture dish, they use ketones fine. If you give brain cells, for example, if you're growing brain cells in a petri dish and you give them ketones, they grow perfectly. And if you take glucose away, they continue to grow and thrive. But if you're growing cancer cells and you take away the glucose and give them ketones, the cancer cells die. And I found this to be fascinating. So we wanted to follow up on that observation. I mean, I wasn't a cancer researcher, but I was really passionate about starting up a project and to follow up with these observations. So our healthy cells have a very high degree of metabolic flexibility. So our brain, for example, and peripheral tissues, think of them as like a hybrid engine, right? So they can use glucose, and under periods of glucose deprivation, they can use fatty acids and ketone molecules. So they use both fuels very efficiently. So cancer cells, they lack this metabolic flexibility due to defects in their metabolism or damage to their mitochondria. So glucose becomes the predominant fuel for cancer cells, and they lack this ability to switch over to alternative fuels. Now, this can be seen that it's been shown that tumor growth is tightly correlated with the level of glucose in the blood. So as glucose levels increase, there's a proportional increase in tumor growth, and several labs have reported this observation. At low levels of glucose, at hypoglycemia, very little tumor growth occurs. An example of this in humans, a great example is a fluorodeoxyglucose PET scan, which shows glucose consumption in tissue. And in this case, this person has metastatic cancer. And you can see areas lighting up in the lung and in the liver and even in the bone with bone metastatic cancer. So what we observe here is that the cancer cells are essentially outcompeting the healthy tissue for glucose availability. And PET scan is really like the gold standard to diagnose cancer, and also to look at the location of cancer, and also to track the aggressiveness of cancer. So the more aggressive and the more deadly the cancer, the greater it's going to show up on a PET scan. So this is an interesting phenomenon, and it's called the Warburg effect. And it was characterized by Otto Warburg, who won the Nobel Prize in 1931. He was the first to observe that these cancer cells have a defect in their metabolism that causes them to ferment glucose for energy. And he was really the first to describe cancer as a metabolic disease. So what he proposed is that cancer cells have dysfunctional mitochondria. And this was a characteristic of all cancer cells. We know that cancer is, if you take a sample of a tumor from an individual or multiple types of cancers, you see a lot of genetic heterogeneity. So there's genetic defects that are very variable between people, and also the same tumor itself. If you take samples from the same tumor, it's very genetically heterogeneous. But pretty much all cancer cells have the same metabolic phenotype, which is this glucose overconsumption. And it's due primarily, Warburg thought, to mitochondrial dysfunction. So if you scan the literature, all these things show up. These are all evident. This is all evidence that there's dysfunctional mitochondria in cancer cells. So I think the greatest evidence comes from electron microscopy studies when we compare normal mitochondria, which have these cristae, which is the inner and outer mitochondrial membrane. This is actually where energy is made. So you create, the mitochondria generates ATP, adenosine triphosphate, within the cristae of the mitochondria. So here's an example of a brain tumor, a glioblastoma, which is a very aggressive kind of brain tumor, and we see a crystallysis, a complete breakdown of the inner mitochondrial membrane. So if this membrane doesn't exist, the mitochondria cannot generate ATP, it can't generate energy. Ketones are metabolized, and fatty acids are metabolized exclusively in the mitochondria. So these organelles need to be functional. You need an inner mitochondrial membrane to be able to develop or generate ATP. So So I know this looks complex, but I'll walk you through it really fast. And just recently, we published an article in Carcinogenesis, which describes that chemical and environmental triggers can damage the mitochondria. So the idea here is that as we age, we get progressive mitochondrial dysfunction, right? So normal mitochondria have very dense cristae, and as we age, the inner mitochondrial membrane starts to break down over time. It's a fact of life that your metabolism declines with age, and there's a progressive decrease in mitochondrial function with time. And the mitochondria generate about 90% of the energy in the cell. So with progressive mitochondrial dysfunction from these chemical environmental triggers, there's a decrease in mitochondrial energy production and a compensatory increase in a glucose metabolism to make energy. So as there's progressive mitochondrial dysfunction from these chemical and environmental triggers, less energy is produced from what's called oxidative phosphorylation. And the nucleus of the cell requires a lot of energy to maintain the fidelity of the nuclear genome, right? So there's very robust DNA repair mechanisms that's constantly repairing damage to your DNA. And these DNA repair mechanisms are intimately linked to energy production. So with mitochondrial dysfunction, you have an impairment of energy production that destabilizes the nuclear genome. This triggers the activation of oncogenes, genes that cause cancer, and can also inhibit tumor suppressor genes, genes that prevent cancer. So oncogenes, the activation of oncogenes increases the enzymatic activity of glycolysis. So it allows cells to generate energy from sugar. So we have mitochondrial dysfunction that causes genomic instability that activates the cancer genes. I like to say this is kind of a controversial theory, but this is what Warburg predicted. And the emerging evidence from various groups are validating this model. So the important question that we ask with that in mind is, could both the organelles be required for the initiation and progression of cancer? So we have the mitochondria's damage, which ultimately damages the nuclear genome, and that kicks on oncogenes. So if all cancers arise from mitochondrial dysfunction, then the replacement of damaged mitochondria with normal mitochondria should prevent cancer. Or in other words, mitochondria producing sufficient respiration, so mitochondria producing ATP, should suppress the growth regardless of the numbers of mutations that are in the nuclear genome. And if that's the case, then healthy mitochondria should be the ultimate tumor suppressor. And we know, just to backtrack a little bit, that ketones, the ketogenic diet can enhance mitochondrial function, and it can enhance mitochondrial biogenesis. So we have more mitochondria. We have mitochondria that are working more efficiently to generate energy. So I want you to know that as I kind of walk you through this, as you think of healthy mitochondria being the ultimate tumor suppressor. We know that normal cells beget normal cells, and that cancer cells beget cancer cells. But if you transfer a nucleus of a cancer cell into with healthy mitochondria, and that cell divides, even though the genetic machinery of this cell is from a cancer cell, the healthy mitochondria suppress the oncogenes in the nucleus. So when this cell divides, it becomes two normal cells. If you take a normal cell nucleus and put it into a cancer cell with defective mitochondria, when that cell divides, even though this is a perfectly normal nucleus with normal genetic information, when that cell divides, it becomes cancer cells. Interestingly, if you radiate the cells and disrupt the mitochondrial function, when that cell divides, it'll become a cancer cell. So this was a series of elegant studies called the Cybrid studies, done by multiple labs, reproduced by several groups. So the mitochondrial dysfunction, just to backtrack, leads to an impairment of energy metabolism from the mitochondria, and this leads to compensatory fermentation, which is essentially sugar metabolism. And the unique quality of cancer cells is that they rely primarily on glucose as an energy source, but they're unable to utilize ketone bodies for energy. So this was the rationale for metabolic therapy, is to exploit the mitochondrial defects that result from the Warburg effect. So the idea here is to establish therapeutic ketosis, elevate blood ketone levels, use a nutrition strategy to decrease blood glucose, glycolysis, and gluconeogenesis. These are all things that the ketogenic diet accomplishes. It also suppresses insulin levels, which drive cancer cell growth, and increase the tissue level of oxygen. So we find that hyperoxygenating tumor tissue increases free radical production, which can help kill the cancer, and it also turns off oncogenes that drive cancer growth. So we asked the question, can therapeutic ketosis and hyperbaric oxygen be used to treat cancer? Now, my colleagues at Boston College and also at Barrow Neurological Institute, Thomas Seifert and Adrian Scheck, have done some elegant work showing that the ketogenic diet can suppress tumor growth in various models. So we wanted to combine the ketogenic diet and hyperbaric oxygen together. And what we demonstrated is that the combination of the ketogenic diet and hyperbaric oxygen can prolong survival in mice with metastatic cancer. So we carefully chose a model of cancer to study that's very aggressive and that relates to the human disease that causes cancer. Most cancer studies are done with an implanted tumor that doesn't metastasize to all the organs of the body. This particular model is very aggressive. When the tumor cells are implanted, there's rapid systemic metastasis to all areas and even the brain, and within about 3 to 4 weeks, the animal succumbs to the tumor burden. Now, we show that with a standard diet and hyperbaric oxygen, there's a delay in the metastatic spread. With the ketogenic diet, there's also a decrease in metastatic spread and tumor growth, but the combination of the ketogenic diet and the hyperbaric oxygen is an extremely effective approach for managing metastatic cancer in this model. We've also demonstrated recently, we published that ketones themselves inhibit cancer viability in vitro. So even under high glucose, if we grow cancer cells, we can stain the dead ones and stain the live ones. And in parallel to that, we grew cancer cells with high glucose and high ketones. And we saw a greater percentage of dead cells in our cell cultures, and we saw a suppression of cancer cell proliferation. So this was even in the presence of high glucose. And we know that ketones can turn off or turn down glycolysis. They also activate various genes that may suppress cancer growth. And we're looking into that now, mechanistically what's going on here. So to take it one step further, we did a combination of metabolic therapy that was the ketogenic diet plus ketone supplementation plus hyperbaric oxygen. And essentially we showed that the combination of these together had the greatest effect overall. And we were able to, so with a control diet, which is high carbohydrate diet, this is a survival curve with that diet. And here's a survival curve with a combination of these things together. Most of the information that I'm going to share with you today was from various foundations that I work with and various consultants that consult directly with people that have cancer and to apply this metabolic therapy. The Charlie Foundation is an excellent organization. They have about 200 clinics across the world, about 150 in the US. And these are two books that I highly recommend if you want to learn more about the use of the ketogenic diet to metabolically manage cancer. So just going back now, just some key points I want to hit on. The standard diet versus the ketogenic diet, when you implement this, the big difference is macronutrient distribution. I think you all know that. With the ketogenic diet, you decrease carbohydrates to less than 10% of calories, so it's very restrictive. And the ketogenic diet is really not a high protein diet, like many people think. It's very moderate to low in protein. So to get into ketosis, there are various ways. There's urine strips or keto strips. Keto sticks are called. You can measure blood ketone levels with a finger stick, similar to a glucose meter, and you can look at breath acetone levels. So these are my ketones on a low-carb diet, and here's my ketone levels on a ketogenic diet that's a little higher in protein than what's used clinically. And this is my ketone levels when I follow a classical, the 4-to-1 ketogenic diet, which is like 85% fat and 10% protein. So I really have to restrict to get my ketones elevated. Now we think ketone supplementation would allow you to achieve this level of ketosis and maintain it with something like a paleo diet or low-carb diet. So we're looking at these things now and studying it in various models. Some challenges to initiation of the ketogenic diet from the feedback that I get from various doctors and consultants that work with patients are liver cancer or elevated liver enzymes. So the liver makes ketones. The liver does not consume ketones. It's one of the few organs in the body that actually can't use ketones for fuel. It lacks an enzyme, succinyl-CoA transferase, and a few other enzymes that prevent the use of ketones for fuel. So the ketones are produced in the liver and they're released into the bloodstream for peripheral tissues. And if you have impairment of liver function, your body is going to not be making ketones very well. Kidney stones or renal disease, these are things that should prevent you from doing this. Kidney stones are a reality with the ketogenic diet. There's a higher incidence, but supplementation of potassium citrate is a way to counteract that. Pancreatitis, fat malabsorption issues, any kind of gallbladder problems, could be problematic with the ketogenic diet. Medications, especially chemotherapeutic agents, can really elevate glucose and prevent you from getting into nutritional ketosis. And various steroid drugs like dexamethasone are often prescribed to cancer patients, and these will elevate blood glucose levels and actually do the opposite of what you want. You can actually feed the cancer. Lack of support within the home. We find that if someone's embarking on trying a ketogenic diet, they really need support from their spouse or their family to kind of eliminate the foods that would be counterproductive to staying in ketosis. High cholesterol is always a concern, and I get emails every day from people that try the ketogenic diet and their good cholesterol goes up, triglycerides go down, blood glucose goes down. So most metabolic biomarkers will go in the positive direction with the ketogenic diet. You may have an elevation in LDL cholesterol, but it's questionable as to why that's, or if that's even important for your health. And food selection can be a big challenge for some people, but I look at it as a good thing because on the ketogenic diet, you can eat high-fat foods like butter and chicken skin and things like that. So I don't see that as a limitation. So there's a variety of studies, 2 pilot studies, one with 16 patients, another with 10 patients. They have demonstrated so far that the ketogenic diet is safe and that it increases the quality of life, and that the outcome in patients, their chances for stabilizing the disease or going into remission correlated with the level of ketosis that they were in. And when I first got into this research, there was about 2, maybe 3 studies on clinicaltrials.gov, and I just took a screenshot of it, and I think there's about 12 or 13 studies. So there's been kind of an explosion of clinical trials starting up, but there's a lot of hurdles to doing this. Funding's an issue, and IRB is an issue, getting approval for these kind of studies. So typically what they do is take the ketogenic diet and they combine it with standard of care, like chemo and radiation. And unless there's like a glioblastoma or advanced cancer where chemotherapeutic agents are largely ineffective. So coming to the end now, there's some questions or topics of discussion, some questions that you may have. Are all cancers responsive? I think maybe cancers that show up really hot on a PET scan would be more responsive to the therapies I talked about today. What dose of hyperbaric oxygen? So this is a very new field. There's the level of depth, the level of pressure, there's how long you should be in the chamber, there's the frequency. Do you do it once a week, twice a week, 5 times a week? We don't know, and that's what we're studying now. Now, the low-carb diet versus the ketogenic diet. And we haven't really looked at a low-carb paleo diet in these models of cancer, but there may be some benefit there. We know the ketogenic diet, you really have to be in ketosis to prevent seizures. So more work needs to be done there. And the big question is, can these therapies be integrated, metabolic therapies be integrated with other therapies? And that's really, to get this into mainstream medicine, we have to think about the ketogenic diet as an adjuvant to the standard of care, because clinicians really are not going to do away with the standard of care, even though it's very limited in efficacy for a lot of things like brain tumors and metastatic cancer. And another avenue that we're looking at is metabolic drugs, things like metformin, DCA. These things are relatively non-toxic, and we think that they will synergize with some of the things I showed you today. And here's some resources that, if you're interested in what I was talking about, there's a variety of resources that I think will be helpful for you in getting more information. And I'm available for any questions. And here's the lab. So our lab, I'd like to acknowledge Office of Navy Research for funding our seizure project that led to the cancer project, and also studying a variety of other disorders, and all the members of our lab that makes this research possible.
Great talk. What about neurological diseases and a ketogenic diet, like, for instance, Alzheimer's, Parkinson's? Have you thought about any hypothesis on that?
Sure. That was actually a topic for an IHMC talk in Ocala. Dr. Mary Newport, who's a personal friend of mine and a colleague, observed that in her husband, when her husband was given coconut oil, which is a source of medium-chain triglycerides, that he improved on cognitive function tests. And so the question is, coconut oil elevates your ketones maybe half of a millimolar. Now we know that ketone esters can elevate that 10 times higher. And the question, I think that's really important, is, well, what do you do when you have, will it be effective if you already have Alzheimer's disease? But I think as a preventative strategy, any strategy that can lower and control blood glucose and help improve metabolic biomarkers like your triglycerides, your C-reactive protein, markers of inflammation over time, would delay, my prediction is that it would delay the onset of these chronic neurological diseases that occur. But I think a lot more work needs to be done. But the work being done so far in animal models and in humans is pretty compelling.
I don't understand why you can eat fat, a lot of fat, and you're not increasing your risk for cardiovascular.
Yeah, that's a good question. That question comes up a lot, actually. So when we eat fat and we restrict carbohydrates, it's forcing our bodies from a glucose-based metabolism to a fatty acid and ketone metabolism if carbohydrates are restricted. So if we eat carbohydrates and fat together, our body's happy using glucose for fuel, so the fat's going to float around in our bloodstream and cause inflammation and cause cardiovascular problems because our body's not fat adapted. Our body's not preferring that fuel source to derive energy from because it's happy, it's getting glucose, right? So the carbohydrate restriction will sort of deplete levels of glucose and lower insulin levels. When you suppress insulin levels, that really enhances fatty acid oxidation in the body. So paradoxically, people who go on a ketogenic diet or high-fat diet see a reduction in their triglyceride levels, which really confused doctors at first. But I mean, for people who study ketogenic diets, it's kind of like obvious, because you're forcing the body to oxidize fat for energy, so it's going to leave the bloodstream and be used by the tissues. And then a high carbohydrate diet increases insulin, and insulin dephosphorylates HMG-CoA reductase. And that's the enzymes that statins. If you guys are on statin drugs, it inhibits HMG-CoA reductase. So that's activated by insulin, which is increased when we eat carbohydrates. So simply eating a lot of carbohydrates will release insulin, and that can drive the production of bad cholesterol. So it's more complicated than that, but that's maybe answer some of your questions.
Time for 2 more questions if
Yeah.
can respond.
Over there? Yeah, right there.
Thank you. That was an excellent talk. The fats that we should be eating, however, are not fats from hamburgers. It should be fats from coconut oil, avocados, walnuts, all your healthy fats. Those are the fats you're talking about.
I'm also, I'm talking about saturated fat too. So the fat that you get from the beef at the store, if it's corn-fed, may have a fatty acid profile that is pro-inflammatory and not very kind of supportive of good cardiovascular health. So I try to select, grass-fed beef and try to go in that direction. So saturated fat has been demonized, and I think it really, the quality and the fatty acid profile of the food you're eating is very important. It's a good point you bring up. Right here.
Okay. So increasing your fat intake and cutting on your carbs, is that the only way, or is there ketones? There's nothing else that you can take to increase the ketones? And just another quick question, is tumors and cancer the same? For instance, you talked about brain tumors, so this would work for those too.
Yeah, so your first question, you can elevate ketones by consuming MCT oil or various products on the market that are ketone salts, essentially, but it's really hard to get them up to the levels that are therapeutic without the carbohydrate restriction. So we're working with kind of synthetic ketogenic agents that may make it to the market or become a medical food in time. So we're kind of working in that direction right now. So your question regarding, is a tumor a cancer cell? So there are some benign tumors that do not express all the hallmarks of cancer, the proliferative capacity, this aerobic fermentation, glucose consumption, and the elevated glucose consumption is really a hallmark of a tumor that's cancerous. So that's why the PET scan is really a gold standard way to, if you see a tumor on an MRI and just a mass, it's really kind of hard to predict whether it's a cancerous tumor. If you do a PET scan and it's lighting up like a Christmas tree, then you know most likely that tumor is cancerous and is malignant. Yeah. Thank you.